YouTube.
Okay, sir. Should
Very
I
good
start?
morning and warm welcome to Ms. Runali Patil, today's speaker. All delegates who are attending this session, my teaching and non-teaching colleague, and dear students. We have been organizing webinar series from academic year 23-24 under the IQAC. And in this series, today we have the first webinar on futuristic innovation in pharmaceutical arena. And this will be continuing throughout the year. We have planned to have a webinar series on every month, and little bit creative idea is that we'll have two speakers. One will be our alumni, and another will be an expert from other areas. And that's why I'm very happy. Today we have my first batch student, Miss Runali Patil, and she is going to talk with all attendees. And this is a very happy moment for a teacher. Our student now has become that competent, can deliver lectures. So this is the first series we have started, and by our college, Gokhale Education Societies, Sir Dr. M.S. Gosavi, College of Pharmaceutical Education and Research, Nasik. As we know that Gokhale Education Society has completed 105 years and is now working with the 106th year, having 140 educational institutes, among that conventional colleges, schools, ITI, technical institutes, management science, engineering college, and this pharmacy college, named after our beloved secretary, Honorable Sir Dr. M.S. Gosavi, that is Sir Dr. M.S. Gosavi, College of Pharmaceutical Education and Research, started in 2012. We have completed 10th year, and within this 10th year, we could achieve many things. Among that, successfully we faced NBA inspection, and ours was the first college got accredited by NBA in 2019. Then we started diploma program, then increasing intake in B Farm. Then we have a two M Farm programs, and not only that, we are also having a research center at our college, and our students are progressing also very lot. Many students are now in a different parts of our globe, and this today's example, Runali Patil, she is working now at Melbourne. Like this, many students are there. They have achieved many academics and professional achievements. And not only students, our staff members also have achieved many things. Among that, 7 staff members visited a different countries, and few staff members have got a best teacher award by society and other organization. And that's why within a span of 10 years, this college is progressing like this. But this year, I'm little bit unhappy that our beloved secretary, Honorable late Sir Dr. M.S. Gosavi, passed away on 9th July 2013, and that was a great loss by our society and for especially this pharmacy college because its name. So it's a request to all of you. We can have a 1 minute silence for this late Sir Dr. M.S. Gosavi. Om Shanti. Now, I'm very sure this webinar series will be continuing throughout the time, and it will be an opportunity for delegate students to hear many experts from different fields. And definitely, this kind of sessions, seminars, workshops, it's help a lot because we can hear the actual experiences, practical problem they are faced and the solution they are achieved, and this could be very very helpful for this. So I'm very happy with this IPSC cell and our college staff member. They have taken effort to start this national level webinar, 1 day national level webinar, and today we have a topic on futuristic innovation in pharmaceutical Arena. And our first speaker is Miss Runali Patil, first batch student of Gokhale Education Society, Sir Dr. M.S. Gosavi, College of Pharmaceutical Education Research. She is presently PhD research scholar at IIT Bombay and working with the Monash University, Melbourne. So this is a very, very happy moment for all of us. Now over to Shilpa Madam. What was it? Yeah.
A very good morning to all. Myself, Mrs. Smita Sharke, Assistant Professor, Department of Pharmaceutical Chemistry. I am very glad to introduce the first speaker of today's first session, Ms. Runali Patil. Her educational qualifications includes uh, basically she is a student, brightest student of our first batch, passed in 2015. She did her post graduation from Pune College of Pharmacy, Pune. Presently, she is a PhD scholar at IIT Bombay in association with Monash Academy, Melbourne, Australia. Her scholastic achievements includes she was worked as visiting research scholar at NCL Pune, currently sponsored with research fellowship by Department of Biotechnology, Government of India. She had 4 publications and 1 patent in her credit. Right from graduation, she started research. Now, she is working on very important topics like development of smart hydrogel for arterial thrombosis, development of trigger responsive block polymer based nanoparticles for arterial thrombosis, and development of gold coated nanobubbles for arterial thrombosis. Today, she is delivering a lecture on advances in stimuli responsive nanocarriers for drug delivery. So, I request Runali to start her session.
Yes. Thank you, Shilpa ma'am. Thank you, Principal sir, for giving me this opportunity to deliver a lecture in such a prestigious series of seminars. Today, I am going to talk about one of the most researched area in the pharmaceutics and drug delivery, that is stimuli responsive nanocarriers for the drug delivery.
Yes, yes. Your presentation is visible, Runali.
Yes. So, today I'm going to talk about advances in stimuli responsive nanocarriers for the drug delivery. As we all know, for the background, that in pharmaceutics we learn, we study, and we research on various conventional drug delivery system as well as some novel drug delivery system. Conventional drug delivery system consists of tablets, capsules, syrups. However, the next step is nanocarrier or nanoparticle based delivery system. But in that also stimuli responsive nanocarriers are the next stage nanocarriers to for a better and effective drug delivery, and also to overcome the drawbacks of conventional drug delivery systems. Coming to the next thing, what is the concept of designing stimuli responsive nanocarriers is that when we design only nanocarrier based system without any stimulus, it leads to the several disadvantages, such as the leakage of the drug or API during the circulation. Also, because of the body protein and various pH changes in our body, these nanocarrier undergoes various chemical and biological changes. Because of that, these become unstable sometime, and leading to the less bioavailability, also rapid clearance. So to overcome these challenges of conventional nanocarriers, we develop stimuli responsive nanocarriers. These includes liposomes, regular solid lipid nanoparticles or polymeric nanoparticles, which includes dendrimers as well as the polymeric micelles. Then some inorganic nanoparticles such as gold nanoparticles or silver nanoparticles. Also, the carbon-based nanocarriers such as carbon nanotubes or the quantum dots. So the concept is we load various APIs or active molecules into these stimuli responsive nanocarriers, and these nanocarriers are designed in a such a way that upon external or internal trigger or internal or external cues, they deliver their API or the drug at the site of disease only when we give them a trigger. Without any trigger, they protect the API or the drug in throughout the circulation, and also it prevents the offsite effects of the API or the offsite toxicities of the API. So this is the basic concept of the designing stimuli responsive nanocarriers. Next is, what are the advantages of these stimuli responsive nanocarriers over the conventional nanocarriers? So basically, these nanocarriers are like a smart rolls. They will only deliver a cargo at the site of disease. That too only when we give them any kind of trigger, either it is external or internal. Because of that, we are achieved to able control drug release without any leakage at offsites. Then also we can improve penetration of the drug in some of the diseases like cancer, where penetration of the drug is very necessary. Then we can achieve a very targeted drug delivery without seeing any kind of offsite toxicities. Then because of that, we can improve the patient compliance with small doses and better effectiveness. And also these kind of systems prevents nanocarriers from the various protein or various bodily changes, leading to enhanced biocompatibility, biodegradability, and also it enhances the cellular uptake. So that's why it is better to call them a smart roles. Next, we will see what are the stimuli or what are the triggers for which we design these nanocarriers. So these are like a smart panda, I would say, and they act on a cue. So these cues can be exogenous or external or physical. That means we can give this stimuli from external outside the body, and these includes ultrasound or light or temperature. And another biological cue is endogenous or internal or biological stimuli. These includes the cues or the triggers which are endogenously present in our body, like pH changes or glucose level changes or oxidative stress or different kind of enzymes. So we will see one by one what are these stimuli. First, we will look on the what are exogenous or external or physical stimuli. I will try to cover maximum examples of the stimuli as I can. So, for what are the exogenous or external stimuli? So, these stimuli we give from the outside of the body. These can be using transducer or voltage cell or magnetic field. So, while these external stimuli includes heat or thermal stimuli, then light stimuli, which includes ultraviolet or near infrared or normal light stimuli, then ultrasound, then magnetic field, voltage or electric field or stretch we can use as a external stimuli. So mostly these stimuli work on the basis of the breaking of bond because of the external heat or external mechanical forces. So we will see some of the examples of these stimuli in coming slides. So, first stimuli is light or photoresponsive nanocarriers. It is like a sunflower, like light makes sunflower happy. These light responsive or light or photoresponsive nanocarriers, they are happy in presence of light or any kind of photo source. So, mostly these stimuli based nanocarrier responds to ultraviolet or visible or near infrared wavelengths. But amongst the all, near infrared wavelength are quite frequently used for designing the photoresponsive nanocarriers, and this is mostly applicable to cancer research or various cardiovascular diseases like atherosclerosis or the thrombosis. In diseases where we need a very quick or very frequent drug release, such as the arterial thrombosis or the myocardial infarction, these near infrared wavelength can be these nanocarriers can be tuned in such a property that they will release very quick they will release the drug very quickly to achieve a maximum biological effect. So while designing these kind of nanocarriers, we use various light sensitive agents which help to give trigger to the nanoparticle, and which leads to the release of the drug. So these light sensitive agents involve various spiroins, then azobenzines, cinnamic acid, and pyrines. Also, some inorganic material or dye, they can be used as a light sensitive agents or photosensitive agents, such as gold nanoparticle based material or gold nanoparticles, or dyes like ICG or IR780. Because these gold nanoparticles or these dyes can also give you imaging properties, these can be used for diagnosis and the treatment purpose simultaneously. That's why we term them as a theranostic agents, which can use for designing the light responsive nanocarriers. So let's have a look on the case study. So this study has been done in our Melbourne based lab, which is at the Baker Heart and Diabetes Institute. So in this study we work on the near infrared right responsive liposomes for the protein drug delivery. So main aim of designing this particle is to treat the bleeding free thrombolysis. In this strategy, we like the authors used thermosensitive phospholipid, that was DPPC, and we used channeling agent, that was breach 58. It also act as a non-ionic surfactant, and to add photosensitive agent, researchers have add the gold nanorod considering their photothermal properties. Here they used the near infrared irradiation at 785 nanometer range, and for 5 minutes using 1.35 watt per centimeter square. From the this 5 minutes treatment, they achieved the protein release about 55.6% in 15 minutes. So in case of thrombolysis, we need quite a quick action. Because of that, we have requirement of such a nanoparticles which can release drug on a quicker basis. So when we give NIR irradiation to the liposomes at the site of thrombus, it released a protein within 15 minutes, which was quite effective for fibrinolysis and ultimately achieving the thrombolysis. So this was one of the example. Instead of gold nanoparticles, we can also use IR780 or ICG dye for quicker release of the drug, so that because of the NIR radiation, the local heat in this nanocarrier will increase, and because we are using the thermoresponsive phospholipids, they will melt with the help of this breach 58, which is non-ionic surfactant, leading to the release of the drug at the site of disease. Then next we will see the next external stimuli, that is ultrasound responsive nanocarriers. So we usually hear, and which most of us have seen the ultrasound machine. So we use it for imaging purpose, but nowadays it is also used for drug delivery purpose, considering it have various properties which can use for the drug release. So ultrasound act mostly a digging deep, like because of its penetration enhancing capabilities and imaging capabilities, we can use it very effectively for drug delivery. So because it is non-invasive treatment, it is highly patient compliable treatment. It improves the better penetration and also enables the spatio-temporal control over the drug release. Mostly our lab works on the ultrasound responsive nanocarriers because we have very sophisticated ultrasound based machines. These can be also used for theranostic purpose, where we can use it for real-time imaging while drug delivering, also in a very low cost and have non-ionizing properties. So the ionizing agents we don't use in ultrasound technique, which make it very safer to use. In this ultrasound, we can use both kind of frequencies: low frequency ultrasound, which is around 100 kilohertz, or high frequency ultrasound, which is above 100 kilohertz, which enables the patient centric use of this ultrasound as a trigger. Then how this ultrasound act as a trigger for this ultrasound responsive nanocarrier? Mostly it's because of its mechanical effects. When we design these ultrasound responsive nanocarriers, we either entrap various gases or air in the nanocarriers, or we use perfluorocarbons in the nanocarriers, which we also called as nanodroplets. These air or these perfluorocarbon make them ultrasound responsive. Because of that, we achieve the acoustic streaming because of the ultrasound pressure, also the shockwave kind of a mechanical effects, and also the cavitation, which is very important mechanism of ultrasound responsive nanocarrier. So when we use or when we give ultrasound to the nanocarriers, they collapses on each other because of the air pressure or the PFC pressure, and because of that the penetration of the drugs increases also. The ultrasound have effect on the cells. Because of the ultrasound mechanical pressure, cell also increases their cell space. Because of that, we get maximum drug absorption at site of disease. Next, we will see the case study. This study is also done in our Melbourne based institute. So in this, the authors, they have worked on the ultrasound responsive nanocarriers, where they developed the N-butyl cyanoacrylate, that is BCA based nanobubbles. So what is nanobubbles? So these are essentially a polymeric particles or the lipidic particles in which we entrap drug and air. Or instead of air, we can use various stable gases such as SF6 gas. Air is very unstable. Because of that, we use SF6 gas in India, but most of the countries in outside India, SF6 is not allowed because of its polluting property. So air stable nanobubbles are essentially very much researched. So here in this study, they have used BSA based nanobubbles, in both way, air stable and SF6 stabilized nanobubbles. So because these particles have air in it, we can see these bubbles are floating at the top of the tube, in this white layer in this third image. um, and also it gives it buoyancy effect. So the very characteristic of these nanobubbles is they float on the surface of the media, unlike the normal liposomes or unlike normal polymeric micelles or polymeric nanoparticles. Here they have used the ultrasound transducer operating at a frequency of 18 megahertz for imaging. So yes, because ultrasound we use for imaging purpose also, they have used this particle as a theranostic particle. And here in the left hand side corner, we can see the imaging done by these particles. B mode, that is burst mode, and CE mode, that is contrast enhance mode, using microbubbles. We can see using the nanoparticles, we have get a better image as compared to non carrier based image. So this particle can be also used as a theranostic particles. And for actual ultrasound trigger for drug release, they have used 1 megahertz frequency with 50% duty cycle. Here duty cycle is very important. Duty cycle means how many times we induce ultrasound waves, or how many times we give ultrasound pulses to the nanoparticle for trigger. So this way they have achieved a very good drug release at the site of atherosclerosis. So basically these particles were for imaging and therapy of atherosclerosis. And they have also used ICG dye for multimodal therapy. So because of ICG dye, they were able to do the IR imaging as well. So this was the case study for ultrasound responsive theranostic particles. Next, we will see the voltage responsive or electric responsive nanocarriers. It's like a keeping the power. This therapy is also non-invasive because we need external nodes or transducer to give the voltage or electric power at the site of treatment. The classic example of this is iontophoresis, which is mostly used for transdermal delivery of the charged compound. So mostly electric field up to 1 volt are used. And even in electroporation is a technique in which we use the cellular space using the high voltage. So in this kind of nanocarriers, we use various conductive materials to synthesize the particles. These kind of materials are either polypyrrole or carbon nanotubes or carbon based nanoparticles like carbon nanocones. These also gives us a better penetration and also the spatiotemporal release over the drug release. We will see the case study of these nanocarriers. So this case study is based on the voltage responsive vesicles or vehicles based on the orthogonal assembly of two homopolymers. So here they have used the polystyrene beta cyclodextrin and the polyethylene oxide ferroquine, that is PUFC. So they have prepared these polymers and using the polymerization technique, and then they assembled it into the particles. So in this study, they have applied the final stimuli plus 1.5 voltage stimuli, uh, whereas they also did the in vitro release using 4 volt, 2 volt, and 1 volt of electric charge. Basically, they got very much higher release of the drug in laser time using 4 volt, but considering the safety, 1.5 or 1 volt is a safer stimuli to obtain the drug release. Then this is because of when we apply the voltage or electric charge from outside, it leads to the changes in the assembly of these particles because of the changes in the assembly of these supramolecular diblock copolymers, and because of that we get the drug release on the cue of this voltage or the electric trigger. So, considering this case study, electric or voltage responsive particles can be a very potential smart nanocarriers. However, the safety and establishing efficacy of these particles in vivo is very important, and this is the next challenge for this kind of vesicles. So, coming to the point, what are the hits and what are the misses of these external stimuli? So, considering the advantages or hits of the exogenous or external stimuli, first we can achieve the controlled drug release. Next, we can improve the penetration of the drug so that we can get maximum effectiveness of the drug. Then we can do the targeted drug delivery using the external trigger responsive nanoparticles. Because of that, we can reduce the toxicity of the nanoparticles. Then we can improve the patient compliance because most of these techniques are non-invasive techniques. Because of that, these are highly patient compliant techniques. Sorry. Then we can also enhance the biocompatibility of and the biodegradability of these nanoparticles by decreasing the various side effects associated with the nanocarrier and leakage of the drug. But there are some misses also when it comes to the external stimuli. The first important miss of these techniques are the tissue damage when we apply external voltage or external ultrasound effect. So because of the mechanical stress and shear stress, we can also observe some tissue damage at the site of the application. So this is the major limitation of these nanocarrier. Then the difficulties in application, although these are non-invasive techniques, but in some in the some area of application, this transducer we can't place directly, or the placing this transducer or the voltaic electrode can be a difficult. So difficulties in application is another limitation, and also the questionable tissue penetration depth. Although we know that ultrasound or applying voltage can improve the tissue penetration, but at what depth we can achieve this tissue penetration is a questionable problem, especially in diseases like the cancer or the endometriosis. We need enhanced tissue penetration. In that case, we can use a biological or endogenous trigger more effectively rather than the external trigger. So these were the hits and misses of the external trigger. Now, based on this background, we will see the endogenous or the biological or the internal triggers. So, endogenous or internal or biological stimulus are like meows who loves to stay indoor. So these include redox, pH, enzyme, or self-regulated system. So basically, biological cues that are present inside our body. We take advantages of these entities to trigger the release from our nanocarriers. So we will see one by one what are these triggers. So first one is redox responsive nanocarriers. Oxidative stress is very prominent in various diseases such as cancer or cardiovascular diseases. In that also, atherosclerosis or myocardial infarction, especially atherosclerosis is majorly arises due to this oxidation and various oxidative moieties like hydrogen peroxide, superoxide ions, then singlet oxygen ions or hypochlorite ions. So mostly these kind of nanocarriers, they are based on the disulfide or thiol bonds, or very so that because of the oxidative stress and GSH, this disulfide or thiol bond gets cleavage, and because of that we achieve the disintegration of the whole nanoparticle, leading to the release of the drug. Alternatively, we can use some redox sensitive materials such as polythioketal or thioether containing polymers, or selenium containing polymers, or boronate ester based polymers. So mostly boronate ester based polymers, they are used for scavenging this redox oxidative stress or singlet oxygen species, so that we can reduce the oxidative stress from the site of disease. Alternatively, these can be used as a trigger responsive material, so that we can release drug at the site of the disease, and also scavenge the oxidative stress, so that we can get the synergistic effect. Next, we will see the case study of this redox sensitive material. So here in this study, they have done the micelle preparation based on the amphiphilic graft copolymer for intracellular delivery of the doxorubicin. So now doxorubicin is used for cancer, so this therapy is for cancer. In these, what are the design strategies? So they have prepared the reduction degradable amphiphilic polyamide grafted polyethylene glycol polymer, which contained a disulfide linkage. So using this polymer, they prepared the micelles in which they load the doxorubicin using the self assembly technique. And when they give the using the self assembly technique, these micelles, they used for in vitro release as well as to check its in vitro effectiveness. So we can see, in presence of DTT, 1 millimolar DTT and no DTT, they have got a highest drug release in presence of DTT. So now DTT is reducing agent, which helps to degrade this disulfide bond, leading to the release of the doxorubicin. So this proves that these kind of nanoparticles can be very much useful and gives a controlled drug release in case of the diseases such as atherosclerosis or cancer, where we need a prolonged and controlled release of the drug for longer period of time. So next stimuli we will see is the enzymes. So these are the enzyme responsive nanocarriers. Mostly the enzymes are present in our body and they do all it wrong, be it digestive process or any other process. So hopping on these enzymes, various researchers have designed such a nanocarriers which can give response to the enzyme and release the drug at the site of the disease. So this is also kind of a disulfide bond or thiol bond based nanoparticle. Um so, yeah. So mostly we will see the case study of this enzyme responsive nanoparticle, so that we will get clear idea of how we design this case study. So mostly in this case study, so the research group have worked on the lipase sensitive polymeric nanoparticles or the nanogels for on demand drug delivery. So in this strategy, they have synthesized the nanogel which are lipase sensitive. So mostly the um bacteria such as S. aureus or any other bacteria, they produce the lipase in larger amount, lipase enzyme in a larger amount. So we can hog this lipase abundancy to remove or to release the drug at the site of infection. So they have designed the triple layer nanogel using the copolymer, diblock copolymer, using the monoethoxy polyethylene glycol and polycaprolactone. So when we injected this drug, when we incubated this drug into the lipase rich media, including these various strains of the bacteria, it leads to the breakage of polycaprolactone chain, and releasing the drug because of the degradation of this nanoparticle. So in this release study, we can see the cumulative release has increased drastically in presence of the 1 mg per ml lipase and ALP, when we incubated into this media, as compared to the without pseudomonas lipase enzyme rich media. So this gives us an insight that um, uh, various enzyme can be used for the trigger responsive drug release, if we design in a such a way that these nanocarriers can be a enzyme responsive. So another classic example of this kind of study is the MMP MMP sensitive nanoparticles for the endometriosis. This example I haven't included in this study, but endometriosis is a very complex disease which require a very controlled and sustained drug release for over 6 months or so. So in this case, nowadays various labs are working so that they can take advantage of presence of MMP9, and they can design such a carriers that they can give stimulus and controlled response to the MMP and release at the site of endometriosis, so that it will be very female patient compliant, and they don't need to take medication in every 15 days or every 1 month. So only 6-month dose is enough for such a kind of nanoparticles. So this was all about enzyme based nanoparticles. um, Next, we will see the self-regulated nanocarriers or self-regulated responsive nanocarriers. So, what is self-regulated based nanocarriers is, there are many systems in our body which can change according to the disease condition. For example, the glucose level in diabetic patient or MMP9 level into the endometriotic patients. So when a person have a diabetes, they can their glucose level can vary very quickly. It can be either very high or it can be either low. It makes that person insulin sensitive, and also we can use glucose level as a trigger for designing nanocarriers. So these systems are capable of responding to the changes in the specific concentration of the analytes. So in these self-regulated nanocarriers, um, we can use glucose level or MMP levels, such a kind of self-regulating system as a trigger. However, the feasibility of this approach is very much questionable. Why? Because glucose level or MMP level in such a conditions is highly variable from person to person. So designing nanocarrier as per the glucose level can vary person to person. So it is very difficult to tune nanocarriers uh, as per the analytes. So this is one of the major drawback of this system, but still it is under research, and we will see one of the example of these self-regulated nanocarriers. So in this case study, researchers have developed the nanosaccharide responsive release of the insulin from polymerosomes of the polyboroxylate block copolymers at neutral pH. So here, researchers have worked on the polymerosomes using the copolymer made up of the boroxyl and the polyethylene glycol. So mostly polyethylene glycol is used to give the steric hindrance effect to the nanoparticle, so that they can be staying in the body for longer period of time, and it will prevent from the rapid clearance of the body. So using these polymers, they prepared different polymeric particles, which include the spherical and cylindrical micelles, as well as the polymeric vesicles, which are spherical in shape. They have turned these nanovesicles as a polymerosomes. So mostly these polymerosomes are designed in a such a way that they will only release the insulin when they will sense the change in glucose or fructose, either it is either in low condition or the high condition. And when they sense the changes in glucose or fructose level or sugar level in the body, or in the neutral pH, they release the drug in a controlled manner. So mostly these studies only till the in vitro release, where they have achieved the variation in the insulin release in presence of sugar and at the neutral pH. But again, the translatability of this approach in vivo or in the clinical trials is very much questionable, and it needs further study in various aspects, also to establish safety and efficacy of these particles for further trials is very necessary. Next, we will see what are the hit and miss of these study of these endogenous trigger responsive based nanocarriers. So as we see in exogenous or external triggers, we can achieve the controlled release and improve penetration of the drug in using this endogenous carrier, also to improve patient compliance. So major advantage of these endogenous cues over the exogenous cues is we don't need to use any external mechanical pressure or external transducer. So it in a way that these systems are very much patient compliant than the external trigger based systems. Then we can improve the biocompatibility and biodegradability of the nanoparticles. But there are some misses or disadvantages associated with these nanocarriers. First one is variation in biological cues. So when we say variation in biological cue, that means like we see self-regulated system, every person's body have different characteristics. Also every person have different biological we say property. So to tune nanoparticles as per the biological cue is itself very difficult task. But we can tune it in a such a way that we can use both biological and external cue. That would be a next step for these stimuli responsive nanoparticles. Then variation in drug release from these carriers, because there is variation of the biological cue. This may lead to the variation of the drug release. So in some instances we may not be able to achieve the controlled release of the drug, also the sustained release of the drug. So when we say that in disease condition like diabetes or endometriosis or atherosclerosis, we need a longer and sustained release, this variation of the drug release can hamper the bioavailability of the drug, which leads to the variation in the effect of the drug. So this is the another major drawback of this system. Then the questionable tissue penetration depth, again this is all related to the variation in the drug release and leading to the variation of the bioavailability of the drug. So how much drug it will get penetrate because of this system is also questionable. So overall these are the hits and miss of the endogenous system. So takeaway message of this system is, although this system is very effective and have some advantages over the exogenous or external stimuli based nanocarriers, there is need to establish more impactful nanocarriers which will be sensitive to even slightest biological cue or slightest analytes like glucose or mmps or any other enzyme or any other analyte we are using as a biological cue. So coming to the next point, we will see the next generation stimuli responsive, where we either mix two of the exogenous stimuli or two of the endogenous stimuli, or one exogenous stimuli and one endogenous stimuli, and we make them more sensitive to the trigger response. So when we say more is better, it is really more is better, and we are using multi-stimuli or multi-stimuli responsive drug carrier for the drug delivery. So we will see the example of it. um, we will see the case study of this. So first case study is the ultrasound enhanced matrix metalloproteinase 9 trigger release of the content from ecogenic liposomes. So when we see ecogenic liposomes, ecogenic means the liposomes which can give have designed carrier in a such a way that they can get maximum and controlled release of the drug. So this is the example of it. So when we say maximum and controlled release of the drug, So they have used these particles will come in contact with MMP9 and will start releasing drugs slowly. And upon demand, they will give the low frequency in the drug, around 50 to 80%, using the 3 megahertz frequency 3 minutes. So that's why, to get a first sustained release of the drug, and later on to get down. So this was the one case study where they have response from mmp9 and ultrasound. Alternatively, we have responsive nanoparticles as well as ultrasound and IR responsive nanoparticles. And they make two modality, or they mix two agents which can use for two different stimuli. So again, this kind of nanoparticle designing requires quite a lot sophistication. So these kind of nanoparticles are next stage nanoparticles. Needs a lot of attention when it is going to the in vivo or further clinical trials. So next, we will see what are the advantages of the clinical translation of the based conventional nanocarrier based nanoparticles. Most products are marketed like liposomes for cancer therapy. When it comes to the stimuli responsive nanoparticles, it's a long way ahead, because only few has been reached to the translational phase. In that also, very prominent is thermosensitive liposomes, because we all know liposomes are very stable and biocompatible and also approved. So here thermosensitive liposomes are used for the treatment of breast cancer, and trials are got suspended because of various issues. So right now these are not in clinical trials. Then iron oxide based nanoparticles, two of them. First one is treatment of cancer, but after 2005 these trials have not been updated and no data is published. So trial and yet to go for long way. And the next one is iron and carbon. These are tested for hepatocellular carcinoma, but again after 2006 and 2007 there is no data published on any of the site for these clinical trials. Major challenges in the clinical translation is first to establish their safety and efficacy. So that we will safe to say that these particles are safe, sophistication in the instrument. So mostly these nanocarriers require ultrasound machines or IR machines. So these available industries see it commercially going forward is a major challenge. So this is the another major, um, we say major drawback of this system. So this was all about clinical translation. So what is the takeaway home message is this technology is very promising, which can overcome the disadvantages of the conventional nanocarrier based system, also improve the patient compliances, especially for the disease conditions which require controlled and sustained release, but also the disease condition which require very quick drug release, for example the thrombosis. But there are a lot of challenges there are a lot of challenges when it comes to the sophisticated tools then its safety and efficacy and commercial challenges. But again, researchers in academia and industry are working very hard to improve these systems and to overcome these challenges. So we will find a way and we will get a better future for this particle, so that we will improve patient compliance and improve the patient's health. So this was all. These are the references for today's presentation. And if any questions, please feel free to ask. I'm happy to answer. And if I can't answer, I will definitely get back to you. So thank you, and you can reach to me on my email ID or my LinkedIn for any queries.
Yes, Runali. Can you see the question on screen? A question by
Uh,
Prakash.
no.
Okay. Can nanoparticles bypass first pass metabolism as compared to conventional medicines?
Yes, nanoparticles can bypass the first pass metabolism, provided that we give them a Trojan horse effect or the steering effect. So we use pegylation mostly to prevent the first pass effect of the nanoparticles.
Okay. The next question is what? What about dose dumping?
Yes, so these nanoparticles we can prevent for dose dumping, because these particles are designed in such a way that these will release the drug only when we will give them a trigger. Otherwise, they will not release the drug. And based on the in vitro data, without and with trigger, we will know what is the frequency of the drug release. So based on that only, we will design a drug in a such a dose, in a such a way that we will not get excessive drug or excessive dose in our body.
Okay. The next question is, can we use in silico technique to understand this?
um I'm not sure about in silico techniques, because to know about the release and response of these nanoparticles, we the only way is to actual check these particles for trigger responsiveness. So, for example, ultrasound based techniques. Mostly we work on ultrasound based techniques. So, like nano bubbles or nanoparticle, they are very quick to the response, right? So I'm not sure how we can use in silico techniques
Okay.
if we don't do it very practically, because nanoparticles can vary their response to trigger based on how what chemistry or what mechanical structure they have. Like I was talking about nanobubbles. So nanobubbles can give quick response, but if we use calcium and alginate based system for same ultrasound, it may give little bit slow response, although it is quick, but as compared to nanobubbles, it will give slow response. So
Okay.
yeah. But if I get to know any other in silico techniques, I have not done it personally. I'll get back to you uh,
Okay.
to
Okay.
the person. Any other question?
No more question. Feedback. Feedback in second session. Okay, feedback link for this session will be given to you in the next session immediately after this.
Yeah.
And all delegates are requested to write their name correctly. They should write along with their affiliation, that is Mr., Mrs., Doctor, name, parents' name, and then surname, like this. Okay.
Yes.
So thank you, Runali, for this informative session. So Ms. Runali has explored the research in the area of gold nano carriers for a drug delivery system. She also explained in detail about the types of stimuli, design strategies, types of nano carriers with case studies. So thank you very much for this session.
Thank you, ma'am, and thank you, the team of Dr. M.S. Gosavi College of Pharmaceutical Research and Education, to giving me this opportunity. Also, giving me various opportunities during my bachelor's day, so that because of them, um, I can do
Please
better and receive.
accept our certificate of appreciation from here.
Thank you. Thank you so much.
Okay, thank you.